Hey guys, how you doing today? Hope you're doing great. So today I'm gonna discuss cell injury part two. Um, in that section, we're gonna discuss disturbances in lipids, carbohydrates, and minerals. I'm Dr. Said Al-Shafi, a lecturer of veterinary pathology um, at College of Veterinary Medicine, Banha University. So let me first start with disturbances in lipids. Before getting into the diseases or lesions um, due to disturbances in lipids, let me first give you an introduction about how the lipid um, being metabolized in the liver, because the liver is a central organ in lipid metabolism. Um, so, the first source of fat you get through the diet. Then you get free fatty acids, or also if you're just burning a lot of fat during starvation or under restricted regimen diet, so you get free fatty acids from adipose tissue, or it's like free fatty acids in chylomicrons in the blood. The end result would be free fatty acids in the circulation, and the free fatty acids go to the liver, and then the liver also has another source of generation of free fatty acids, and this is through conversion of acetate into fatty acids. The fatty acids in the liver, um, most of the fatty acids will be converted to triglycerides, and this is by the action of alpha glycerophosphate. And this process called esterification, but the minority portion will be converted into other products like cholesterol, esters, phospholipids, and ketone bodies. So the majority will be converted to triglycerides. This is very important. And then what happens, there is um, like lipid acceptor protein or apoprotein. This will bind the protein to triglyceride to make lipoprotein. And then lipoprotein goes to the circulation and then goes to the organs that needs that. So this is a mechanism uh, or the pathway of lipid metabolism in hepatocytes. So the first lesion we're gonna say is lipidosis or fatty change, um, or steatosis or fatty metamorphosis, same condition. So steatosis is mostly used by physicians, doctors, and as you see, whenever you say fatty change, it seems like it's an organ where the you should not see fat in it. And then what ended with is there is fat appearing in the organ, and this is kind of the lesion or abnormality you're gonna see in the organ. So it's um, it's very important to understand every single term you're studying, because maybe from the definition you can tell the story behind it. So let me get to the definition. So lipidosis is accumulation of triglycerides and other lipid metabolites um, within the parenchymatous cells. It's very important to understand that it's the accumulation of fat occurs inside the cell. It's intracellular in the parenchymal cells of the body. Okay, and then since the fatty change uh, always happens in the liver, most of the textbooks say lipidosis, like mostly correlated with hepatic lipidosis. So most of the textbooks would mention hepatic lipidosis, since most of this lesion happens um, in the liver. And since also the liver is a central organ in lipid metabolism, but also occurs into the heart and skeletal muscles and kidneys. So the etiology, any abnormality in this pathway would lead to fatty changes. So, for example, if there is excessive delivery of free fatty acids from any source, like either diet or adipose tissue, will end with more free fatty acids, more triglycerides, and then fatty change. Or decrease beta oxidation of fatty acids to ketone bodies and other substances, and due to toxins or hypoxia. Like if there is decrease of conversion of free fatty acid to products like cholesterol, esters, phospholipid, and ketone bodies. This means there is increase in conversion to triglycerides, and then leading to lipidosis. Or impaired synthesis of apoprotein. This happens in carbon tetrachloride toxicity and aflatoxicosis. So in these conditions, usually like carbon tetrachloride toxicity lead to deficiency of apoprotein, which is very important of in binding of triglycerides to the protein and then creating lipoprotein. Rare conditions like impaired combination of triglycerides and protein to form lipoprotein, or impaired release of lipoprotein from hepatocytes into the circulation. So this slide is more detailed than the other slide in terms of etiology, as it correlates the lesion with diseases most common. So first one lead to fatty change is conditions that cause an increase in mobilization of body fat store, and this happens in two important diseases. Um, the first one is pregnancy toxemia, and this happens in late pregnancy, and ketosis, and this happens in early lactation in dairy cows. Also, any nutritional disorders like obesity, for example, um, where there is increase in dietary lipids, or there is a great mobilization of adipose tissue to the liver. Or protein calorie malnutrition. This means like decrease in proteins, and apoprotein wouldn't find a protein to bind it to triglycerides, and with less production of lipoprotein and accumulation of more triglycerides in the liver. Starvation also the same. Like there is increased mobilization of triglycerides in that condition. Or it may be secondarily to an inherited disease like Wilson disease, and also like in diabetes mellitus, the same thing. Or poisoning by chemicals, like carbon tetrachloride and yellow phosphorus toxicity, and we discussed that before in the previous slide, because this lead to deficiency of apoprotein, which is very important in creation of lipoprotein. Also, in some disorders, like feline hepatic lipidosis, and sometimes named as feline fatty liver syndrome and fat cow syndrome. And in these two conditions, the cause of hepatic lipidosis is not clear and it's not known. So, how the fatty change looks like from outside. How I know this liver has fatty change? So grossly, if there's mild fatty change in the liver, you wouldn't be able to know it. Basically, the liver would be looking normal. But if the liver has marked lipidosis, so the liver is always enlarged, yellow, soft, and friable. And how you gonna know it's soft and friable? You can force your finger into it, and if it make hole, you you know that it's soft and friable. Um, the edges of the lobes are rounded and broad. So whenever you see the lobe, so this surface, it should be sharp and flat. But in fatty chain, you're gonna see it more rounded. If you consider this is the end of the loop, you're gonna see the border kind of rounded, not sharp. On incision, in the normal liver, if you force a knife here into the liver, so in normal liver, you're gonna see the edge of the wound you're gonna make by cutting the surface to be sharp and flat. The edges will be sharp and flat. But in fatty liver, you will see that the cut surface will bulge and swollen like this. And this is because the capsule was very stretched because the liver was enlarged, and the capsule dragged the border towards the outside, and then this is what happens. It kind of bulging whenever you do a cut into the surface. The hepatic parenchyma is still soft and friable and has greasy texture uh, in fatty liver. Also, if you cut 1 centimeter section from the liver and you put it into formalin or water, this will float. So this section will float on the surface, and this is also a distinction and characteristic of fatty change compared to other conditions you would know, hydrophobic degeneration and glycogen infiltration. We're going to discuss glycogen infiltration in a way in the next few slides. Um, so just a reminder, um, it's important to distinguish the gross lesions from the lesions present in glucocorticoid or steroid hepatopathy in the liver and in dogs, mostly like glycogen and water. And this will discuss that there is two conditions that are similar to fatty change, um, and those are glycogen and water deposition. Um, in those two conditions, the liver is enlarged as well and has also rounded edges, but the color is different. So um, in these two conditions, usually they are pale. The liver is pale, beige to tan white, somewhat firm, um, and doesn't have grazy texture or grazy cut section. And the cut section do not the cut sections do not float in the formalin. So these are the differences, if you would, between fatty change and glycogen infiltration and water deposition. So under microscope, how fatty chains looks like. The hepatocytes of lipidosis or hepatocytes with lipidosis um, are mostly vacuolated, and the extent of vacuolation indicates the severity, which means more vacuoles mean it's more severe. Less vacuoles, it's mild. So initially, um, this is how it starts. Initially, there are few small clear vacuoles in the cytosol or the cytoplasm, and those vacuoles later would increase in size and number and even coalesce into larger vacuoles, as you see here. So the small vacuoles would coalesce, make large vacuoles, and will increase in number as well. And then they can even coalesce these large vacuoles into only one large vacuole that occupies a whole cytoplasm and push the nucleus towards the periphery and making signet ring appearance. So, so these vacuoles um, have sharp delineated border. So you can identify um, the border because it's very clear. You can tell, oh, the end of the fat um, is very clear, so you can tell this is a fat and this is the rim of cytoplasm and hepatocytes. And this represents the hydrophobic interface between water and lipid in the cell cytoplasm. As I mentioned before, hepatocytes, um, so the vacuoles in the hepatocytes can fuse together and just make one big, large vacuole that can push the nucleus towards the periphery and make signet ring appearance as you see. In extremely cases, the hepatocytes can be filled with lipids that in this condition you can't even know um, where are the normal hepatocytes. You wouldn't see normal hepatocytes, and the liver would be like just a fat um, replacing most of the hepatocytes. And the only things that you can identify at that condition is presence of portal areas as indication of the liver. So then here we're gonna say that not every single vacuole you see in the hepatocyte would be fatty change or fat deposition. So as we mentioned before, there are two conditions where you can see some vacuoles in the hepatocytes. And those are glycogen infiltration and water deposition. But you can, in order to identify which vacuole you're seeing under microscope, um, basically you have some special stains for glycogen, if like periodic acid shift, if it's a positive, it's a glycogen, or you can use special stains of fat like Sudan 3, and this stains the fat yellowish orange, or Sudan 4 or scarlet red, and this stains the fat orange to red, like here, or osmic acid or osmium tetroxide, and this stains the fat black in color, as here. Or night blue sulfate, and this stains neutral fat, oils, and cholesterol esters red in color, and fatty acids and phospholipids blue in color. And oil red O stain, and this stains hepatocytes. I'm sorry, stains fat red in color. So the second lesion we're gonna discuss is fatty infiltration, and it's different than fatty change, that the one that we discussed before, because in fatty filtration, this condition occurs in an organ that has some fat stored in it under normal condition. Um, but fatty change, it happens in an organ, normally doesn't have fat. Um, Also, in fatty change, the fat deposition is inside the cell. So it's intracellular. In fatty infiltration, it's extracellular. It's away from the cell. So it's just deposition of fat outside of the cell, usually in connective tissue. So you should not be confused between the two terms. Normally, the adipocytes are present in connective tissue, and this is even reflects the nutritional state of the animal. Um, so the fat usually present in fasciculi, skeletal muscle, and between cardiac muscle subepicardially. And the fat usually stored um, in that area. Whenever the adipocytes increase in number, this process is a fatty infiltration that we're discussing now. Mostly, uh, it happens in old age and obesity conditions, um, where there is hyperplasia, which means increase in number of adipocytes due to proliferation of pre-adipocytes. Um, so in this condition, um, some myocytes may undergo atrophy and then disappear and then replaced by adipocytes. If there's generalized excessive infiltration of neutral fat in depot and connective tissue of parenchymatous organ, this disease will be like the obesity, reflect of obesity condition in that case. Then the third lesion we're going to discuss in disturbance in lipid metabolism is cholesterol deposits. Some textbooks mention cholesterol deposits under the term lipoidal degeneration, and just to get through the cholesterol deposits, we know that most of this condition happens in condition like atheromas, where there is deposition of fat into tunica intima of some arteries, um, and then somas as well. Okay, so the cholesterol crystals, um, whenever it's deposited into some organs or into the wall of arteries, um, the body will deal with it like as a foreign body. Then inflammatory cells will go to that area to get rid of it. Um, but what happens is there is settlement of chronic inflammatory cells around the cholesterol crystals, and this makes the granuloma um, or granulomatous inflammation. Sometimes the granulomatous inflammation or accumulation of chronic inflammatory cells around the cholesterol crystals, named as cholesteatomas, and this mostly seen in a plexus of old horses. If you looked at it in the three dimensions, the crystals are thin, rhomboidal plates with notched corners, and mostly seen in an area of hemorrhage or necrosis. Grossly, usually it is seen as uh, friable pale yellow nodules in the tissue. Um, Under microscope, cholesterol crystals are dissolved out um, during the histological processing, and this is by the action of xylene material that gets rid of all the fat basically inside the cell. Um, but anyway, by if the crystals are dissolved during the histological processing, it leaves a circular cleft or needle-shaped cleft, as you see here, kind of needle-shaped inside the tissue, and you can see aggregation of chronic inflammatory cells around the clefts here. Mostly it's macrophages and giant cell, as you see. Then we're gonna discuss disturbances in carbohydrates metabolism. So the condition here is glycogen infiltration. Before discussing glycogen infiltration, you have to know that glycogen is normally present and stored in hepatocytes and myocytes. And this is kind of a reserve energy um, for the body if needed. Hepatocytes of starving animals. So if there is a starvation, um, so see, like during the starvation, glycogen will be consumed by the body. So you should see that the cells would be devoid of glycogen in a few days of starvation, basically because the glycogen should be consumed at that time. If you saw some glycogen left over, um, this represents or reflects glycogen infiltration because it was excessive infiltration somehow. Um, and some conditions like in diabetes, glycogen is not only found in hepatocytes, but also in kidneys and beta cells of islets of Langerhans. So mm, the etiology would be like a disturbance in glucose or glycogen metabolism, and this most common in diabetes or genetic disorder, um, glycogen storage disease, sometimes named as glycogenosis or dextrinosis, um, in which there is a deficiency in glycogen metabolic enzyme or the enzyme that break glycogen basically to be used for energy. Or excessive corticosteroid administration. This will also lead to glycogen infiltration. Um, normally, I mean, normally, glycogen should be um, increased in young growing animals, and animals in that condition are reflected by well nourishment. Basically, the animals that have more glycogen means they are well nourished. Grossly, um, if there is physiologic deposits of glycogen or normal amount of glycogen, you wouldn't be able to tell whether the liver has glycogen infiltration or not. But if there is massive glycogen deposition, like an steroid-induced hepatopathy, the liver will be enlarged and pale in color, and also has rounded border um, in that condition. Under microscope, the glycogen would appear as a clear vacuole in the cytoplasm of the cell. Um, it's um, it looks like clear spaces with irregular outline and distinct outline. So the outline of the vacuole is not clear like in fatty change that we discussed before. So just a reminder, in fatty change, usually the fat inside the hepatocytes will be either rounded or ovoid, but in glycogen will be irregular mostly. And the border in the fatty change is sharply delineated and clear. Here it's indistinct border. Usually in the fatty change, you see the nucleus peripherally located, but here the nucleus mostly is centrally located. So the nucleus remains centrally located in glycogen infiltration, as we know, but in very rare condition, um, these vacuoles can push the nucleus towards the periphery as well. So to know the difference between fatty change and glycogen infiltration or water, you can use special stain as we mentioned before. So the glycogen has some special stains like periodic acid shift. This technique stains the glycogen purple to magenta, as you see here, or pistis carmine, and this stains glycogen bright pink in color. Pathological calcification. Um, we can discuss disturbances in minerals metabolism. Pathological calcification. So, what is pathological calcification? It's a deposition of calcium salts. Whenever I say salts, means maybe calcium plus phosphates or carbonates or magnesium or iron. Um, like usually, calcium is combined with other minerals. Um, so the deposition of calcium salts in dead or dying tissue or normal tissue. And if the depositions in dead tissue would have a name, and if it's normal tissue would have another name. Okay, so the forms of calcifications. The first one is dystrophic calcification, and the position here, if you, in order to remember it, so this consider like death. So dystrophic calcification means like the position of the calcium in dying tissues, despite of normal serum of calcium. So what that means is calcium deposits into tissues that underwent death, and the calcium level is normal. Metastatic calcification is second one. Metastatic calcification is the deposition of calcium salts in normal tissue due to hypercalcemia, due to increasing calcium level in the blood. So the first two are the most common, and most of textbooks would mention uh, those two. The third one, and this is rare, idiopathic calcification, which means um, you don't have either um, dying tissue and you don't have hypercalcemia. So this is usually named as idiopathic because you don't know the etiology or the cause. So occurs in absence of tissue injury or abnormality in calcium and phosphorus metabolism. Iatrogenic calcification, the fourth one, is calcification that happen due to an error an error in um, like giving oral solution that has calcium to the animal. So introduced calcium to the animal, or injection that you resulted into remaining of calcium in a certain site. So basically, this is a calcification you introduced from an external source or from your end. Like this is ingestion of calcium salts from calcium chloride containing solution, for example. Then, what is dystrophic calcification? Dystrophic calcification is the deposition of calcium salts in dead or dying tissues. And it occurs in areas of necrosis. Necrosis means death of the cell. It occurs in all types of necrosis except, or it's minimal, I would say, in liquefactive necrosis. The mechanism to the dead or dying cells can't regulate the influx of the calcium into the cytosol, and calcium accumulates into the mitochondria and in two phases we're going to discuss in the next slide. But basically, the dystrophic calcifications um, happens in certain sites. For example, in necrotic myocardium or skeletal muscle, or granulomas. We discussed granuloma before, like cholesteatomas. Um, Granuloma is the aggregation of chronic inflammatory cells and giant cell, such as tuberculoid granulomas in cattle, and maybe dead parasites, such as hydatidus in cattle and trichine in pigs. Um, so if the parasite um, died into a certain organ, calcium can deposit it on this parasite. Maybe also old thrombus or tumor that underwent necrosis and then calcification happens. Also occurs in calcinosis. Usually calcinosis, it's a calcification into the skin, and I'm going to discuss this in the next slide. So calcinosis is a calcification in or under the skin. And there's two forms of calcinosis: calcinosis cutis and calcinosis circumscripta. Calcinosis cutis, um, there is mineralization of dermal collagen and epidermal and follicular basement membranes. And it happens in areas where local skin local skin damage has occurred. You can see here like there is damage in this area, and this lead to calcinosis. And calcinosis circumscripta mostly associated with um, repetitive trauma. Um, and this is, for example, like a site of buried sutures of polydexanol, sutures that got inserted into the tissue, and for certain reason you didn't remove it, and it remained in that site. So this usually suture would make repetitive trauma at that site and lead to necrosis in that area and then calcification later. The deposition of calcium um, usually occurs at the bony prominences where bone and skin um, thinly separated, um, or in food pads and mouths. This is, for example, food pad or paw. Um, there's also classification in that area due to repetitive trauma in that case. Okay, here we got pathogenesis of calcification in general. So there are two steps. The first step is initiation or inoculation, where calcium released from mitochondria and then um, bind to or adhere to the phospholipids of plasma membrane and other organelles. But by release of calcium, this will activate phosphatase, which break phosphate. Then calcium sat free and phosphate sat free. Then calcium will bind with the phosphate, form calcium phosphate. And this cycle um, repeats many times, and more calcium forms, microcrystals develop due to some structure changes, and then this lead to calcification basically because of accumulation of microcrystals. And this process occurs in intracellular in mitochondria of the cell or extracellular where the calcium get from outside into a vesicle, then released in the cytoplasm, and the cycle happens again. Usually, intracellular occurs in dystrophic calcification, and extracellular occurs in metastatic calcification. The second form of calcification is metastatic calcification, and definition is the deposition of calcium salts in normal tissue secondary to hypercalcemia. So the mechanism here is the entry of large amount of calcium ions into the cell due to hypercalcemia, and then these calcium ions will precipitate on organelles, particularly mitochondria. And we discussed that before into the pathogenesis, if you remember the two phases, initiation and propagation. So before getting into the causes of metastatic calcification, just a reminder for how calcium level basically is regulated into the body. Um, so there is two hormones basically control um, calcium level in the blood. Um, the first one named as calcitonin, and this is released from thyroid gland. Second one is parathyroid hormone or parathormone, and this is released from parathyroid gland. Calcitonin, its main function is to get the high blood calcium level um, decreased, then lead to normal blood calcium. In other words, if there is high um, if there is high level of calcium into the blood, calcitonin will take this calcium, put it into the bone for making more bone. Basically, the calcium um, get into the bone will be used by osteoblasts to make bone. And then by taking care of the high calcium in the blood and put it in the bone, usually the calcium level return to the normal um, condition. So adjusts the level of the calcium. Okay, then if on the other hand there is low decrease in calcium level in the blood, parathyroid hormone has to deal with it. What happens is parathyroid hormone um, activates the osteoclast, which is the bone-eating cell, to induce lysis of the hydroxyapatite of the bone. So it will lyse some bone matrix, so the calcium released into the circulation, and then once the calcium released, it will adjust. the low level of the calcium and return to a normal um, level in that case. So calcitonin take care of high blood calcium level um, and return it to normal by sending the calcium to the bone. Parathyroid hormone take care of low calcium level by lysing or lysis of bone and get calcium into circulation and lead to normal calcium level at the end. What is the etiology? First, first is parathormone and PTH related protein disturbances like primary hyperparathyroidism, for example, or hypercalcemia and elevated PTHRB, and this happens in canine malignant lymphoma and adenocarcinoma of Purkinje gland of anal sac. Mostly, the calcium deposition would occur in intestinal mucosa, vessel walls, lung, kidneys, or destruction, excessive destruction of bone due to primary or metastatic neoplasm, like Paget disease or immobilization or tumors as multiple myeloma and leukemia, for example, or metastatic neoplasm like breast cancer, for example. Or it's due to renal failure. So in renal failure, usually there is retention of phosphates, and this induces secondary hyperparathyroidism, and then lead to hypercalcemia. The sites will be calcium salts deposition mostly in gastric mucosa, kidney, and alveolar septum. Or vitamin D toxicosis due to ingestion of carcinogenic plants like Sistrum diurnum by herbivorous, and usually the deposition would be into the aorta, heart, and lungs, or due to ingestion of rodenticides containing cholecalciferol, and this happens in dogs and cats. The sites will be intestinal mucosa, vessel wall, lung, and kidneys. Okay, then how we how we gonna know there is calcification in the organ? Grossly, the affected areas of the tissue would be looking white or gray, like you see here in myocardium. There is white area in the myocardium, and this is represent calcification anyway. And on incision, you're gonna have a gritty feel and sound. So you're gonna see as this sound, gritty feel or gritty feel when you cut, or gritty sound basically, um, like a sand or something. When you scrub a sand on a floor, this make gritty sound. On a microscope, the calcium stains blue with methicillin and appear as a fine amorphous granules or clumps. For example, this is a clump of calcium and it's blue in color, and can be either intracellular or extracellular, inside the cell or outside the cell. The full extent of calcification can't be determined by H&E stain slide. You can't tell how extensive is the calcification by H&E slide, and you can use a special stain like Von Costa stain, and the calcium here appear black. And you see, this is Von Costa stain, um, and appear black, or there's a green red S, and the calcium appear red in color. Okay, um, just notably, the Von Costa stain is not specific for calcium, contains also the phosphates and carbonates, but these substances always in combination with calcium. So the last lesion I'm gonna discuss in disturbances in minerals metabolism is a heterotrophic bone or ectopic bone. It is a process of production of bone at abnormal site. There are two types of ectopic bone. Heterotrophic bone, and usually it happens due to like some embryonic cell settled and in an area um, that not supposed to go. For example, like if an embryonic cell that make the bone settled and in the area where the a liver is being formed. So you can see like a spicule of bone inside the liver. Um, and this found at many sites like spicule or nodule of bone into connective tissue of lungs of dogs and cattle or dura mater um, in dogs and at sites also dystrophic and metastatic calcification. Osseous metaplasia is the second type, and this is the most common one. And and mm, from from the term itself, you can say meta means a conversion, right? Plasia, you can consider it like a cell. This is conversion of the cell from a certain type to another type. So here, as a fibroblast will be differentiated into osteoblast. Fibroblasts that make connective tissue will be converted to osteoblasts that make bones, and this results into formation of osteoid into normal tissue. So, for example, you can see this is a kidney, and then this is glomeruli, and this is a osteoid that was formed into the glomeruli, and this named as osseous metaplasia. This is the end. Thank you so much, and looking forward to making other videos. Please share and subscribe. Thank you so much.